Hey Beaks, it's Mr. Boylan, and today I'd like to draw out mitosis with you. As you can see, I use red, blue, and yellow pretty much, and something to write with, pen or pencil, or I'm using a Sharpie here today. And before we really start talking about mitosis, we should start at the beginning, and that is the cell cycle. The cell cycle is the ordered events that every cell has to go through. Okay, and you can break it down into 4 phases. Okay, we have G1, S, G2, and M. And as you can see, M is only about 10% of the cell cycle, and that's what we care the most about today, right, is M phase. You can lump together G1, S, and G2. Okay, these 3 phases together make up interphase. And if you know what the prefix inter means, it means between, right? So these things all happen in between cell division. Okay, M phase is actually when a cell divides. Okay, but G1 is actually a misnomer. It says first gap here. Scientists, when they first looked through a microscope and said, wow, the cell doesn't really look like it's doing a whole lot here. Oh, sure it is, right? It's a brand new cell. It just divided, and it's going through so many reactions. And metabolism is very high, and it's basically acting like a factory. But you can't really see a lot happening under the microscope. But boy, oh boy, a busy cell right here. S phase, that stands for synthesis, right? And that's when you copy your DNA. And then G2, that's the second gap. Again, it doesn't look like there's a lot happening, but sure there is. Remember the surface area to volume ratio, and sometimes cells are getting triggered via these little extracellular signals, such as like a cyclin, and it's telling the cell, hey, it's time, let's divide here. So G2, you're actually getting ready to divide, and then M, yeah, that's what we paid to see, right? M phase, okay, is when the cell divides. And when you say the cell divides, you have to be careful because that's broken into 2 parts, mitosis and cytokinesis. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm. So this is actually like your cell splitting. This is your nucleus splitting. Okay, so this stuff's important, right? A lot of kids, when they learn this for the first time, are thinking like, blah, blah, yeah, it's boring, it's boring. This is where a lot of oncology research is, right? This is what we're trying to study to fight cancer. A lot of times it's just little hiccups in the conversation that happens here. The guys that tell us how to do this get a little messed up. And if we could just find out, well, why is the conversation not working the correct way? Boom, you have a treatment for cancer. So cell cycle, very, very important stuff here. Okay. All right, next I'd like to talk to you about chromosomes. Okay, now inside your nucleus, where eukaryotes, right, so we have this nucleus, we have, the double helix. And the double helix is a macromolecule. It's DNA, all wound up, A, T, C's, and G's. You have a sugar phosphate backbone, right, nitrogenous spaces in between. But you'll run into these 8 proteins called histones. And DNA has a way to wrap all around. This is, again, not to scale by any means, but these histone proteins are in there, and your double helix wraps all around them, So eventually, DNA forms this thick fiber. It's not just double helix. It's double helix and protein mixed in, and this is what we call chromatin. Now, chromatin is what's sitting right now in our nucleus, and we have a bunch of it, right? If we were in a G1 cell, remember, we had a mom and dad that each gave us 23 chromosomes, so we have 46 pieces of chromatin inside of our nucleus. These would be like gigantic pieces of spaghetti, okay? If the double helix is kind of like thin fishing string or something like that, then the big thick spaghetti noodles, that would be like your chromatin. And you have 46 pieces. After S phase, right, you copy your DNA, you would have 92 pieces of this chromatin. Huge amounts. That's why cell division is so amazing. We're going to take these huge pools of spaghetti noodles or chromatin and find a way to perfectly separate them to each side of the cell so we can divide it. Pretty awesome stuff. So what happens is these chromatin fibers, they eventually start to coil up, okay? And there's videos I'll show you when we learn more about this. And they coil and they coil and they supercoil and all this. So, by the way, when the double helix wraps around these histones, that's something that we call a nucleosome. So now you have all these nucleosomes in here and everything's wrapping, and then it wraps again and again and again. But eventually you're going to have this beautiful structure that we call a chromosome. The amount of DNA that's mixed into there, lots and lots and lots and lots of DNA, okay? But we had to do this. Imagine if I gave you a plate of spaghetti with all these huge noodles all over it, right? And there's 92 of them, and I said, hey, do me a favor, separate them perfectly, really quickly. Well, that would be really hard to do. So this is almost like taking a selective fork and saying, oh, I'm just going to coil up this noodle right now. And then another fork saying, I'm going to coil up the same one. Okay, and they come together. That's the beauty of cell division. You had to take all these huge amounts of DNA, and the first thing you do is you coil it up, we'll see. So when you see a chromosome, okay, let's draw it like this here. It has two parts on either side, and this is going to be very important when we talk about genetics later and things like that, but this is, or these are called sister chromatids. Okay, these sister chromatids are genetically identical. Okay, now we'll get back to that in a second. Maybe this chromosome right here, let's just pick one. Okay, maybe it's like number 7 from my dad. Okay, and now let's draw another chromosome. Let's draw number 7 from my mom, maybe. Okay, so this is the exact same chromosome right here, only this one is from my mom. So these are my chromosomes right here. Okay, but one of them was paternal. Okay, this came in the sperm. Okay, one side of it did. And then this one came in the egg from my mom. That's maternal. Together, these chromosomes are what we call homologous. Okay, this is a homologous pair right here. So homologous chromosomes means that the genes are the same. Okay, so I'm just going to write same genes. Now what you're going to learn with genetics is it could actually be something though that has different alleles are possible. Okay, and all that means is this. Okay, my dad, for example, my dad had free earlobes, and actually I don't remember if this is on chromosome 7 or not, the gene for earlobe attachment. But the bottom of my earlobes, if you've seen them, oh my gosh, I look like Dumbo. I have huge, huge earlobes. So my dad gave me what we call a dominant form of a gene there, and wherever the gene is, let's say the locus for a gene is right here, meaning the double helix that's in here has letters or a sequence that gives me my free earlobes. So my dad, okay, gave me big E, is what we'll say when we get into genetics. That's a form of a gene or an allele. My mom, at the exact same spot, though, my mom has attached earlobes. Okay, and she gave me a different allele. It's the exact same gene. The genes are the same on these, but she gave me a little e, little e. Okay, so the same genes, but the alleles are different. So I right now then, if you saw my ears, I told you, you would see that I have free earlobes. They're very big, right? And that's because I have a big E, which says, yeah, go ahead and have free earlobes. But I also carry this little e from my mom, meaning my kids, I could either pass free earlobes to my kids or attached. And it would depend on what my wife does, right, to see what my kids would look like. So let's go back again. Sister chromatids are genetically identical. And the reason that this makes sense, right, you can actually think of it as S phase. Right? We copied our DNA in S phase. So this used to be one piece of chromatin, right? And during S phase, or during DNA replication, we took this and exactly copied it. So that eventually when we form chromosomes, it would look like this. Okay. So basically, this and this have to be genetically identical. Whatever my A, T, C's, and G's, the sequence of DNA is on this chromatid, has to be the same as this one. Now over here, they're probably very similar, but you could have different alleles. Okay, so in this case, I have the attached earlobe allele for the earlobe attachment gene from my mom. So that's why these are homologous. They're not identical, these two chromosomes. The sister chromatids are, of course, identical on each one of them, but the homologous pair, they're different. Okay, and sometimes they might be the same. For example, let's talk about, who knows, gene Y. And maybe my dad gave me, you know, big Y, and my mom gave me big Y, big Y. Okay, whatever. Sure. Now these are still homologous, but in this case, they actually have the exact same allele. Up here for the earlobe, they have different alleles. It doesn't matter. Okay, so sister chromatids, genetically identical, same sequence of DNA. Homologous chromosomes, oh, same genes, okay, but they don't necessarily have the same alleles. They could, but they don't have to. Okay, another area that you should know between these is where it's held together. Each chromosome, okay, has an area of DNA that we call the centromere. All right. So the centromere is a section of your chromatin. Okay, when it coils up, this is in the middle where the two sister chromatids will come together. Okay, and that's pretty much what a chromosome is. So next thing we have to find out, well, when do we coil these up and how do we separate them? Okay, so here we go. We're going to talk about how cells divide. And I'm going to take a cell right now that's pretty much right at the end of G2 here. We're right at the end of interphase, and we probably have enough cyclins. And if you read ahead, maybe they connected to this maturation promoting factor. They formed a new molecule that's a trigger, basically. And this trigger says, hey, we're ready to divide. Let's go into M phase. Okay, and G2, let's call it actually late G2, late interphase. Our cell is probably pretty big, right? Remember that surface area to volume ratio is pretty important. And I am not going to draw all the stuff inside the cell here. And I'm going to just draw, like, in other words, I'm not going to draw the rough ER, the mitochondria, things like that, because they're not super important for today. But of course, they're all in there, right? Everything's there that should be there. But what I'm really going to draw is, first of all, I'm going to draw my mom's chromosomes. At this point, if you're good, you know that my mom gave me, 23 chromosomes, but they've already copied themselves. Oof. 46 pieces of DNA in there from my mom, 46 pieces of DNA from my dad. Holy cow, do I have a lot of stuff going on in there. Okay, so those all exist right now as chromatin. We didn't do anything with them yet. I probably have this little dark area in there, okay, and we'll see what that is in a second. I have a nuclear envelope, right, or envelope if you're weird, and that's going to surround the nucleus, so there's a membrane. That's what makes us eukaryotic. And also inside of my cell, I probably have, I usually use yellow for these, a centrosome. And then just try and draw that a little bit yellow. I don't know if that'll show up well or not. I just usually make like two little lines here. So here we go. Let's label what's going on here. First of all, I have the nuclear envelope. This little dot that I drew, that's the nucleolus, right? That's where you're making your ribosomes. Inside of the nucleus, that's where we have our chromatin. Didn't coil up yet, right? And then down here, this cloud of cytoplasmic material, okay, this is called a centrosome. And what's inside of there are, remember when we learned animal cells, we said, oh, animal cells have centrioles. And that's what's inside of the centrosome, with a pair of centrioles. Okay, so this cell went through S phase already. We're now in G2. We're getting ready to divide. Let's see how we do that. One thing I want to talk about, though, and this will be much more important when we do meiosis, but a human being is what we call a diploid cell. And what that means is when my dad gave a sperm during fertilization to make Mr. Wonderful, me, that sperm had 23 chromosomes in it. So we call that N, or the haploid number is 23. And then my mom's egg, okay, which we'll talk about why that's so much bigger and cooler, but that had 23. So these are both haploid cells. And when fertilization happens, that makes something called a zygote. And because now we have both sets of chromosomes together, we call this not a haploid or a haploid cell anymore. A zygote is actually a diploid. So it's actually going to be 2n equals 46. So what this means is a haploid cell means one set of chromosomes is 23 total. One set of chromosomes is 23 total. But as a human, we have two sets of chromosomes because we all have a mom and a dad, and that gives us 46 total. I don't want to draw 46 chromosomes. Okay, that would take me forever to film this video. So what we're going to do is we're going to draw a cell that is a 2n equals 4. Okay, so as we keep going, we're just going to draw 4 chromosomes. But in the back of your head, remember, we actually have 46 of these if this was a human. Okay? All right. So that is G2 late interphase, and we're about to start M phase. Okay, so here we go. We're done G2. We're now into M phase, or the mitotic phase. And for our current text that we're using, there's 5 phases. A lot of times you just say prophase, metaphase, anaphase, telophase. Prometaphase, some textbooks like to put in between here. This is kind of like late prophase, okay? But it is pretty different, so it's good that we differentiate there. So let's start our mitotic phase here with prophase. Okay, here we go. So I have that same cell that we just left off with, okay? But what happens here is, it's pretty awesome. The chromosomes, of which I'm going to draw 4, they actually start to coil up, right? The chromatin fibers that I was talking about, it's almost like somebody took their fork and is starting to really make them loop back on themselves. So in the nucleus, my mom's number 1 starts to coil up quite a bit, okay? And my mom's number 2 starts to coil up. All right, and these are pretty stringy at this point. They're really just starting to. Here's my dad's number 1. So that's also going to start to coil. And then my dad's number 2 is starting to coil up. All right. Now, what else is going on here? Well, the nuclear envelope actually started to break down a bit, right? It's starting to come apart, okay, mid to late prophase. Another thing that happens is that centrosome copied itself. We actually started to make something that's really cool. Okay, so we have these 2 little like cylinders in here. Those are the centrioles. And the centrosomes, they start to shoot out these microtubules all over the place. This is called an aster right here. It kind of looks like a starburst. But in between them, these microtubules start to grow towards each other. Okay, and if you remember microtubules, we talked about them before with cilia and flagella, the dining arms pulling on them, things like that, or maybe tracks for organelle movement. And I said, you know what, wait until later in the year. You're going to see just how important microtubules are. And here's why they're so important. These things are going to really help rip our chromosomes apart. So what is the hallmark of prophase? Like if I was looking through a microscope, the last cell, how would I know that I'm now in prophase instead of in G2? Oh, yeah, chromosomes coil. Right? Chromosomes coil. When I see that, I know for a fact, oh yeah, yep, this is definitely in prophase right now. And of course you have the centromere here in the middle, right? So the centromere is in the center there, and this is made a chromosome made of 2 sister chromatids, genetically identical. And again, this is a 2n equals 4 cell. I'm not going to draw 46 chromosomes. 2n equals 4. But normally there'd be 46 chromosomes in there for a human being. Okay, but I'm not going to draw that. So that's prophase. Step 1 to making a 2 little baby daughter cells. All right, here we go. Okay, here we go with prometaphase. Now, a lot of people just consider this, like I said, late prophase. So if your book doesn't talk about prometaphase, you could just consider it later prophase. Okay, and a little bit different from the last picture. Here, our nuclear envelope is pretty much gone. There might be a fragment here or there, but that thing's pretty much out of the picture at this point. The next thing is the centrosomes. They're pretty much at opposite poles of the cell right now. So they have those centriole pairs inside of them, and they're pushing microtubules out all over the place, right? But the microtubules here have really started to come out towards each other. All right, so what exactly are they grabbing? Well, the chromosomes are now very fully coiled. Okay, so here's my mom's number 1, right? And maybe here's my mom's number 2 down here. And let's see, here's my dad's maybe number 2 over here. Oh, let's call that number 1. That's pretty big. And here's my dad's number 2 maybe over here. Okay. Now, how are these things connecting to these microtubules? You have these little proteins on either side of the centromere, okay, where the spindle can grab here. So these little proteins are what we call kinetochores. Okay, so that's what the mitotic spindle is going to grab. And there's other spindle fibers that aren't grabbing anything, right? And they're just reaching out. So this is a prometaphase cell or a late prophase cell because the nuclear envelope is gone, right? The mitotic spindle that's forming, okay? In prophase, it was an early mitotic spindle. Now it's a later mitotic spindle. It actually grabbed the fully condensed chromosomes, and it's actually starting to move them towards the center of the cell. So as this microtubule grabbed the kinetochore here, this chromosome is heading this way. Okay, and these chromosomes are heading this way. They're heading towards the center. All right. So what's the hallmark of prometaphase? I would say there's no more nucleus, right? Or the nuclear envelope is gone. The chromosomes are fully coiled. They're connected at their kinetochores to the mitotic spindle, and they're starting to head towards the middle of the cell. All right. Let's do the next phase. Okay, metaphase is pretty simple. Actually, it's the quickest phase out of all of M phase. So here's our cell, same thing, 2n equals 4. We're just drawing 4 chromosomes. But now the mitotic spindle is fully formed. So we have our centrosomes on either side. And I believe we had mom's number 1 up here and mom's number 2 somewhere down around here. Okay, and then we had dad's number 1 way down here, and dad's number 2 up here somewhere. Okay, so we have our centrosomes, and inside of there are centrioles, and of course they're shooting out microtubules, and sometimes they head out right in the right direction. So here we go. We are connecting our microtubules to the kinetochores again. Okay, that's what a metaphase cell looks like. And again, you would have 46 of these things lined up. But in this cell, we just have 4. And they line up right down the equator of the cell. Okay, this is an imaginary line that we call the metaphase plate. Okay, and as you can see, what happened here are the spindle fibers grab the chromosomes in prometaphase. They move them towards the center. And when you're at metaphase, that means, yep, you are finished. That's why it's such a quick phase, because nothing's really going on here. It's just you did it. You lined all of the chromosomes up right down the center of the cell. Okay, so that's metaphase. So the hallmark really is just lined up along the metaphase plate. Okay, let's go to the next one. Okay, here we are with anaphase. This cell, or this phase of mitosis, got a little famous from the movie Twilight. The vampire, and they were in biology class looking through the microscope, and anaphase was in there. So for you twihards, there you go. Anyway, this is a pretty cool phase. So I'm going to draw the cell a little later in anaphase. So I'm going to draw it a little bit more long than circular like we've been doing. And again, we have our centriole pairs, of course, in our centrosomes that are separated here. But now something interesting happened. All right. My mom's number 1 used to look like this, right? It used to be the chromosome connected, and we're about to split it. So my mom's number 1 chromosome, half of it went this way and half of it went this way. My mom's number 2, half of it went this way and half of it went this way. It's getting pulled toward the centrosomes. My dad's number 1 is getting pulled this way and getting pulled this way. My dad's number 2 getting pulled that way and that way. Okay, so let's fill this in and talk about this. This is pretty important. So I have my cylinders in there, these centrioles inside my centrosomes, and as you know, we're connected to the kinetochores. Okay, so the kinetochores are still here, right? And we're now moving these things toward, not towards the center anymore, we're going towards the poles. And that's the hallmark of anaphase. The second you split a chromosome, you started anaphase. Now there's something, it's really just, biologists have to name everything, but this one's very important. We just talked before about how this right here, my mom's number 1, this is a chromosome made out of 2 identical sister chromatids, right? So very good, and if you know that, you're doing well. However, in anaphase, the second you split them and they come apart from each other, you no longer call them sister chromatids. These are now called daughter chromosomes. And you might find that annoying, okay? And it kind of is that the name changes there. But the thing is, these are about to be their own chromosome inside of a brand new cell. So calling them a sister chromatid wouldn't make any sense because their sister is gone. It doesn't even exist in that cell. So the second you split these things up, you now have what we call daughter chromosomes. Okay, no longer a sister chromatid. Okay, so why, this is a little bit late anaphase. Anaphase begins right here. As soon as they come apart, that's the start of anaphase. When does it end? It ends very late when they stop moving. What I drew here is about midway, maybe a little later in anaphase. Why is the cell looking a little bit longer, like an egg? Well, remember how some of these spindle fibers were not connected? Okay, and you have tons of these fibers. The ones that aren't connected, they don't know that, oh, I'm supposed to be heading towards the pole right now. All the chromosomes are heading towards the poles, but the spindle fibers that aren't connected, they're still growing towards the center. And what happens is, when these non-kinetic or microtubules hit each other, it starts to lengthen or elongate the cell. Okay, so technically, this is kind of like the beginning of cytokinesis. We're almost starting to separate the cytoplasm a little bit. Okay, so again, hallmarks of anaphase, I would say, well, the chromosomes were lined up down the center in metaphase, and as soon as they split, boom, now you're in anaphase. And what do you call them once they split? Daughter chromosomes. Okay, and why is the cell getting a little bit longer? Well, the non-kinetic or microtubules, they're starting to run into each other, and when they do that, it pushes the poles apart. Okay, one left. Here we go. Okay, we did it. We got this far, and now it's time to talk about telophase and cytokinesis. So this is the end of M phase, and once we're finished here, we'll be right back into G1. So now I'm going to draw this same cell, only now it's starting to pinch in the middle. Okay, and what is happening here is, remember how the cell got longer, and that's because the non-kinetic or microtubules were pushing against each other. So your cell got really long. Now what happens in here is, you remember your cytoskeletal fibers, you have one called actin, okay, these little microfilaments. It's almost like pulling your hoodie strings, right, to close them. There's a ring of actin right here that's starting to pinch, and what we do is we call this the cleavage furrow. So the cleavage furrow is in between what's about to be 2 brand new little cells. And if you remember what we brought to this side, over here, well, we had my mom's number 1, but you know what? It started getting a little bit unraveled again. We also have my mom's number 2 in there, right? And that's starting to get a little bit more unraveled. We have my dad's number 1. Oh yeah, and my dad's number 2. Okay. And the same thing over here. We had my dad's number 1, my dad's number 2, and it's starting to get unraveled, folks. Oh boy. So we're going back into, there's my mom's number 1, my mom's number 2. We're heading back towards becoming chromatin, right? We had those beautiful spaghetti noodles all wrapped up along those forks so we could separate them very easily. But now we're done separating them. Hey, let's let them go back to being just spaghetti, right? And another beautiful thing happens here. Guess who comes back? Ah, the nuclear envelope starts to reform, right? So it's the opposite. This phase is very much the opposite of prophase. You might see your nucleolus show back up, right? So, looks like a crazy person's eyes. But what's happening here is, yeah, you're setting yourself back up. So you look kind of like prophase, right? So you're reforming the nucleus, your daughter chromosomes, right? Your full-fledged daughter chromosomes that used to be sister chromatids, but now that they're here, they're daughter chromosomes, they're starting to uncoil back to chromatin. Your nucleolus is showing back up. And yeah, sure, you still have your centrosomes in there, one per cell here, right, with our centriole pairs in there. But the microtubules, they stop now production. They don't really need to do much more. And eventually what's going to happen is this cell is going to split here. Now here was the beauty of this, and this is what's crazy. We started with a 2n equals 4 cell, right? Well, wait a minute. Mitosis is basically asexual reproduction. How can we have identical cells when we're all finished here? Well, think about it. We used to have, when we started, 4 of these things, and these are called chromosomes. And now we have 4 of these things in each cell. But guess what these are called? Chromosomes. So technically, we started with 2n equals 4, and they were double-stranded chromosomes, and now we have 2n equals 4 in each cell. 2n equals 4, 2n equals 4. Oh, it just means that we have 4 of these. But guess what? S phase will eventually happen, and after you go through S phase and then G2, you can make these all over again. So you're right back to where you started anyway. Okay? So telophase is really just you reform the nucleus, and now what's going to happen here? Well, these cells eventually split, and we have these cute little baby daughter cells. And in this cell, I have chromosomes from my dad, and in this cell, I have chromosomes from my dad. Maybe this is my skin cell. When I was in high school, I played football and baseball and things like that. Maybe I got cut sliding into third base or something on a triple. Yeah, right. I was not fast enough for that, but here we go. Maybe I was making a tackle at linebacker and busted my elbow up, and my cells all of a sudden had to replace themselves. Well, this is how I do it. Asexual reproduction was mitosis, right? And each of these cells now has 4 chromosomes, just like what we started with, and they are genetically identical. So we've created 2 genetically identical daughter cells. Cool. And what phase are both of these in right now? Hey there, little cell. I'm G1, G1, right? And I just split. My surface area to volume ratio feels really good. I'm pretty efficient, and I'm just doing my thing now, and I'm going to go through that cell cycle and continue. Now, this was an animal cell. I do want to show you one more thing. Plant cells do it a little bit differently. One second. Okay, so one last thing here. Cytokinesis, like we just saw, is the division of the cytoplasm that has to happen along with mitosis, right? So you divide the nucleus and the cytoplasm to make your new daughter cells. And in animals, it's easy, right? Animals, like we just saw, cleavage furrow, we split them in half, and no problem there. The problem is plants, right? Plants have cell walls. So cytokinesis is very simple. When you get to telophase in a plant cell, which is the last phase of mitosis, think about what's happening there. Okay, so plants had one parent give DNA, the other parent gave DNA, just like in us, right? I mean, it could be the same plant. That's called self-pollination. But point is, they have 2 sets of DNA. Their nuclear envelopes started to reform. They're eukaryotes, right? Very important. And what's going to happen here, though, is this. Because they can't pinch, because, yeah, they have a plasma membrane, don't forget. Right? There's a plasma membrane right underneath that cell wall. But because they can't pinch like we can because of that cellulose cell wall, what they do is they form a cell plate. Okay? This is cellulose and some other proteins and sugars that start forming in the middle here. Excuse me. So this is a cell plate starts to form. And it's basically cell wall material. And what's going to happen is eventually these vesicles that you form here, they're going to fuse. So this would be like a telophase plant cell, probably at the bottom of the root or something like that, or in a leaf that's forming, going through mitosis. And eventually when these things all fuse, oh, look what we just did here. We actually just formed 2 plant cells. And what phase would these both be in? Well, these would now be in, once that cell plate fuses to form a cell wall, these would both be in G1. So cytokinesis is a bit different, excuse me, in plants because they can't pinch like we can. They have to do it a different way. So they basically just build a new wall in between. Okay. And that's pretty much the phases of mitosis. So I hope you guys found that helpful.